Good day and welcome to another segment in human histology. I'm Dr. Louisa Ko, with you for the next half hour or so for the lecture on histology of the immune system, the lymphatic tissues, and organs. The immune system is the physiologic capacity of the body to defend itself against microorganisms, pathogens, foreign substances, and transformed cells, while the lymphatic system is its morphologic counterpart. The components of the primary lymphatic organs include the bone marrow and thymus, where cells of the immune system called the lymphocytes are born and educated and released upon maturation into the secondary lymphatic organs. The secondary organs are made up of diffuse lymphatic tissues, nodules, lymph nodes, spleen, and others. It is here that in the presence of an offending antigen, the lymphocytes are activated to become immunocompetent effector B and T cells, able to differentiate between self and non-self and initiate the appropriate response. An antigen is any substance that induces a specific immune response and may be in the form of a pathogenic microorganism, environmental hazards, or transformed cells that mimic foreign cells like cancer cells. The body reacts by mounting either a specific or non-specific immune response. Non-specific pre-existing immunity refers to defenses that are innate to the body, such as the skin and mucous membranes, constituting physical barriers against foreign invasion, low pH levels that contribute to chemical defenses, enzymes in body fluids that neutralize foreign cells, neutrophils, monocytes, and phagocytic macrophages, and natural killer cells, along with mast cells, basophils, and dendritic cells. The next line of defense comes in the form of adaptive immunity, providing specific and immune memory after initial contact with an antigen. As part of the innate arm, neutrophils identify pattern recognition receptors, or PRRs, on bacterial surfaces to trigger an immediate immune response within minutes to hours of exposure. They are the main cells recruited in the initial stage and trigger phagocytosis and destruction of the antigens, followed by macrophages. These cells are then drained through the lymph into the lymphatic tissues to result in adaptive immunity through the activation of T and B lymphocytes. Specific immunity is further categorized as being humoral or cell-mediated, depending on the response mounted against a specific stimulus. B lymphocytes are the primary effector cells in humoral response and work by producing antibodies that neutralize toxins, viruses, and bacteria and mark them for destruction by other immune cells like macrophages, or the activation of the complement system, which enhances the function of antibodies and macrophages. They are primarily responsible for outside cells that enter the lumen of body tracts, as well as those in the extracellular fluid. T lymphocytes mediate cellular immunity through specialized antigen receptors that recognize antigen peptides presented to them in the MHC, or major histocompatibility complex molecules of antigen-presenting cells. This is especially important against viral, fungal, and mycobacterial infections, as well as against tumor cells and in understanding transplant rejection. The cells that make up the immune system primarily consist of three major lymphocytes: the B cells, named after the bursa equivalent organ, the bone marrow, where they are derived, T cells after the thymus, the natural killer cells, which develop from the common lymphoid progenitor, and the supporting cells. Approximately 70% of lymphocytes are immunocompetent cells circulating in the blood or lymph for immunologic surveillance, while the remaining 30% migrate into tissues lining the respiratory, gastrointestinal, urogenital tracts, and the skin. In order to fully understand the next topics, let us get to know CD and MHC molecules. CD, or cluster differentiation molecules, are surface markers expressed at different stages of lymphocyte differentiation, which can then be used as immunohistochemical markers to identify and quantify them for clinical use. For instance, CD2, 3, 5, and 7 are all present in mature T cells, but the presence or absence of CD4 and CD8 subclassifies them as either helper or cytotoxic T cells. Major histocompatibility complexes are molecules that serve as peptide displays on antigen-presenting cells for T cell recognition, either of foreign or self-proteins. MHCs allow antigens to be recognized by T cell receptors, or TCRs of lymphocytes, when presented to them by APCs. MHC1 is found in all nucleated cells and platelets and present endogenously synthesized short peptides of 8 to 10 amino acids to cytotoxic T cells. In essence, they serve as targets to signal elimination of only abnormal or infected host cells. In contrast, MHC2 are only found in antigen-presenting cells and present larger exogenous peptides of 19 to 20 amino acids long for CD4 recognition. Cytokines are substances secreted by activated T cells that act on lymphocytes to affect the immune system processes. Similar to these, growth factors are biologically active molecules that affect somatic cells. Interleukins are secreted by helper cells and promote T, B, and hematopoietic cells. Here is a table of important interleukins that play a role in hematopoiesis and immunity. As we go along the next few subtopics, you might find it useful to reference this table to recall their various functions. We now move on to the effector cells of the immune system. T cells account for up to 80% of circulating lymphocytes and are responsible for cellular immunity. This table summarizes the actions and roles of different T lymphocytes. Helper T cells express CD4 markers that are designated Th1 cells. They secrete gamma interferon, interleukin-2, and tumor necrosis factor, cytokines that activate macrophages, which in turn phagocytose bacteria and other organisms. Th2 helper T cells synthesize interleukins 4, 5, 10, and 13, which initiate IgE, mast cell, and eosinophil-mediated responses that destroy extracellular parasites. The third subset of helper cells, designated Th17, synthesize interleukins 17 and 22, which recruit neutrophils in bacterial and fungal infections, but also increases defensins that help with epithelial barrier function. Cytotoxic T lymphocytes express CD8 markers that are activated on exposure to antigens and inject cytotoxic proteins into abnormal cells, including those in viral and parasitic infections and cancers, and plays a role in acute transplant rejection. This diagram illustrates the activation of helper T cells after exposure to antigens. CD4 cells recognize antigen peptides expressed on the surface MHC2 of antigen-presenting cells, which trigger the production of the specific lymphocyte needed for the pathogen. The diagram below shows the action of cytotoxic T cells. T cell receptors recognize MHC1 antigen peptide complex on infected cells, which trigger the release of cytotoxic molecules that lyse and kill it. Regulatory lymphocytes suppress immune responses by influencing the activity of other cells in the immune system. For example, the CD4, CD35, FOXP3 lymphocytes. Gamma delta T lymphocytes comprise only 5% of T cells and are of the subtype that migrate into epithelial tissues of the skin and mucosa of various lumen and stay within the interfaces of the external and internal environment, serving to encounter antigens on the surface of epithelial cells before they enter the body. Mucosa-associated invariant T lymphocytes express invariant alpha and beta chains that recognize riboflavin metabolites that occur in fungi and bacteria, secreting inflammatory cytokines to destroy infected cells. They present a second line of defense against organisms that enter the portal circulation and evade lymphocytes of the GI tract. B cells account for almost 30% of circulating lymphocytes and produce immunoglobulins. They also express B cell receptors that serve as antigen binding sites, which, when activated, initiates the humoral immune response. They also express major histocompatibility complex II molecules that present antigens to T cells. This table summarizes the different antibody isotypes produced by the human B cells, where they bind to, and their functions. Immunoglobulin G is the main antibody in secondary immune response and is responsible for activating the complement system, stimulating chemotaxis, and is capable of crossing the placenta, causing passive immunity. IgM, on the other hand, is the main antibody in secondary immune response, responsible for fixing complements, activating macrophages, and serve as antigen receptors for B cells, along with immunoglobulin D for mature B lymphocytes. Immunoglobulin A is the primary antibody in body secretions, while IgE is responsible for anaphylactic reactions and is predominant in parasitic infections. This is the schematic representation of an antibody. The isotype is determined by the type of heavy chain present. The fragment antigen binding, or FAB fragment, binds to the specific antigen, whereas the crystallizable FC fragment fulfills the B cell effector functions such as complement activation. Effector cells express FC receptors on their surfaces, which anchor antibodies at their FC fragments. Although comprising only close to 15% of circulating lymphocytes, NK or natural killer cells are an integral part of the innate immunity and are often called large granular lymphocytes. They are regulated by inhibiting natural cytotoxicity receptors or NCRs on natural killer cells. Like cytotoxic T cells, natural killer cells are programmed to recognize transformed cells and release perforins and granzymes that lead to the lysis of targeted cells. In the primary lymphatic organs, the bone marrow and thymus, lymphocytes are programmed to recognize a single antigen, and they enter the blood as immunocompetent, albeit naive cells. These cells, along with plasma cells, organize around reticular cells and fibers to form effector tissues, such as the lymph nodes, tonsils, and spleen. In these secondary or peripheral lymphatic organs is where they undergo antigen-dependent activation into effector lymphocytes and memory cells. Inflammation is the nonspecific response of the body to pathogens and may be to either sequester the invasion, digest the antigen, or to degrade it. If a macrophage phagocytoses and degrades an antigen, it triggers immune recruitment and presentation of the antigen MHC2 molecule to immunocompetent helper cells to start specific immune response. The initial contact with an antigen is usually initiated by B lymphocytes in a few days and is mostly composed of IgM. Proliferation and formation into plasma cells occur, and some memory cells remain in circulation in anticipation of reinfection or re-exposure. Because of the presence of previously sensitized memory B cells, the secondary immune response is faster and stronger, characterized by the predominance of IgG antibodies. This reaction is responsible for anaphylaxis and is the basis for the science of immunizations. Type 1 anaphylactic hypersensitivity occurs rapidly, happening within minutes at re-exposure. Previously sensitized mast cells and basophils release histamines, proteases, eosinophil chemotactic factors, leukotrienes and interleukins, and amplified by platelet activating factor, which is responsible for the vascular manifestations of anaphylaxis. This is mediated by IgE antibodies and are neutralized by eosinophils. Recall from previous slides that for activation of the immune response, there must be an initiating event. This first signal occurs when the T cell receptor of helper or cytotoxic cells interact with the antigen MHC complex on the antigen-presenting cells. Remember that cytotoxic or CD8 T lymphocytes are MHC1 restricted, and helper or CD4 T lymphocytes are MHC2 restricted. A co-stimulatory second signal happens when the membrane molecules of both cells interact. One important interaction is between the B7 molecule on the antigen-presenting cell and CD28 on the T cell membrane. However, as a regulatory mechanism, a competitive inhibitor